Hello dear students, welcome to this program. Today we are discussing an interesting topic which is molecular biophysics, forces involved in biomolecular interactions, Ramachandran plot, dihedral torsional angles, supercoiling of DNA, linking, twisting, writhing, and how ligands interact with biomolecules. Let me start with our first objective, which is molecular biophysics. Molecular biophysics is a rapidly growing field of research that plays an important role in elucidating the mysteries of life's molecules and their assemblies, as well as the relationship between their structure and function. Molecular biophysics is the science that combines mathematics, physics, chemistry, and biology techniques to determine how living organisms function. The questions posed by physicists are, for example, how does the brain process and store information? How does the heart pump the blood throughout the body? How do muscles contract? How do plants extract light energy in photosynthesis? While biologists, physiologists, and geneticists work toward answering the same questions, biophysicists focus on the physics and physical chemistry of the processes. The questions apply to various levels of complexity and structural organization. On a large scale, biophysicists study how organisms develop and function. At a smaller scale, they investigate individual organs or tissues, for example, the nervous system, the immune system, or the physics of vision. Other groups quantify processes such as cell division that takes place within single cells. Finally, at the finest level of organization, molecular interactions are analyzed via sophisticated experimental and theoretical techniques that overlap the areas of genetics, cell biology, biochemistry, and molecular physics. Biophysics is the study of the physics of certain complex macromolecular systems, cells, and organisms that function under conditions of insignificant temperature and pressure changes. An organism can be thought of as an intelligent, self-controlled chemical machine that is self-regulated by molecular signals, molecular receptors, and transducers of information. The basic biological function subsystems are nucleic acids, biopolymers, peptide chains, proteins, and specialized proteins called enzymes. Biophysicists seek to understand biophysical processes by accounting for intramolecular and intermolecular interactions and their resulting electronic and structural conformational changes, and by studying the transfer of electrons, protons, metallic ions, and energy within biological systems. The hierarchical organization of biophysics is. Coming to our second objective, which is what are the forces involved in biomolecular interactions. The life of a cell depends on thousands of chemical, biochemical interactions and reactions exquisitely coordinated with one another in time and space, and under the influence of cell's genetic instructions and its environment. Biochemical interactions are of two types: covalent interactions. Second one is non-covalent interactions. Covalent interactions. Strong forces form a covalent bond when two atoms share one pair of electrons, which is called a single bond, or multiple pairs of electrons which form double bond or a triple bond, etc. Second one is non-covalent interactions. The weak attractive forces of non-covalent interactions are equally important in determining the properties and functions of biomolecules such as proteins, nucleic acids, carbohydrates, and lipids. There are four major types of non-covalent interactions: ionic interactions, hydrogen bonds, van der Waals interactions, and the hydrophobic effect. Electrons are shared unequally in polar covalent bonds. In many molecules, the bonded atoms exert different attractions for the electrons of the covalent bond, resulting in unequal sharing of the electrons. The extent of an atom's ability to attract an electron is called its electronegativity. A bond between atoms with identical or similar electronegativities is said to be nonpolar. In a nonpolar bond, the bonding electrons are essentially shared equally between the two atoms, as is the case for the most carbon-carbon and carbon-hydrogen bonds. However, if two atoms differ in their electronegativities, the bond between them is said to be polar. One end of a polar bond has a partial negative charge and the other end has a partial positive charge. Covalent bonds are much stronger and more stable than non-covalent interactions. Non-covalent interaction. First, ionic interactions. Ionic interactions are attractions between oppositively charged ions. Ionic interactions form the attraction of a positively charged ion, a cation, for a negatively charged ion, an anion, NaCl for example. The bonding electron contributed by the sodium atom is completely transferred to the chlorine atom. In aqueous solutions, simple ions of biological significance such as sodium, potassium, calcium, magnesium, and chloride do not exist as free, isolated entities. Instead, each is hydrated, surrounded by a stable shell of water molecules, which are held in place by ionic interactions between the central ion and oppositively charged end of the water dipole. Most ionic compounds dissolve readily in water because the energy of hydration, the energy released when ions tightly bind water molecules, is greater than the lattice energy that stabilizes the crystal structure. Hydrogen bonds determine water solubility of uncharged molecules. A hydrogen bond is the interaction of a partially positively charged hydrogen atom in a molecular dipole, example water, with unpaired electrons from another atom, either in the same intramolecular or in a different intermolecular molecule. In the strongest hydrogen bonds, the donor atom, the hydrogen atom, and the acceptor atom all lie in a straight line. Non-linear hydrogen bonds are weaker than linear ones. Still, multiple non-linear hydrogen bonds help to stabilize the three-dimensional structures of many proteins. Hydrogen bonds are both longer and weaker than covalent bonds between the same atoms. Van der Waals interaction. Van der Waals interactions are caused by transient dipoles. When any two atoms approach each other closely, they create a weak, non-specific attractive force called a van der Waals interaction. These non-specific interactions result from the momentary random fluctuations in the distribution of the electrons of any atom, which give rise to a transient, unequal distribution of electrons. In two non-covalently bonded atoms are close enough together, electrons of one atom will perturb the electrons of the other. This perturbation generates a transient dipole in the second atom, and the two dipoles will attract each other weakly. Similarly, a polar covalent bond in one molecule will attract an oppositively oriented dipole in another. Van der Waals interactions involving either transiently induced or permanent electric dipoles occur in all types of molecules, both polar and nonpolar. Now coming to the hydrophobic effect, which causes nonpolar molecules to adhere to one another. Nonpolar molecules do not contain charged groups, possess a dipole moment, or become hydrated. They are almost insoluble in water, that is, they are hydrophobic. The covalent bonds between two carbon atoms and between carbon and hydrogen atoms are the most common nonpolar bonds in biological systems. Nonpolar molecules or nonpolar portions of molecules tend to aggregate in water owing to a phenomenon called the hydrophobic effect. Because water molecules cannot form hydrogen bonds with nonpolar substances, they tend to form cages of relatively rigid hydrogen bonded pentagons and hexagons around nonpolar molecules. This state is energetically unfavorable because it decreases the randomness or entropy of the population of water molecules. Nonpolar molecules can also associate, albeit weakly, through van der Waals interactions. Now the third objective, which is the Ramachandran plot. A peptide bond is basically a chemical bond which is formed between two molecules when the carboxyl group of one molecule reacts with the amino group of other molecule with the elimination of the elements of water. The peptide bond has a considerable double bond character, and this prevents rotation around the bond in the polypeptide chain. Adjacent amino acids can adopt different configurations by rotation around the two other bonds in the backbone. The angle of the bond between the nitrogen atom and the alpha carbon atom is represented by symbol phi, and the angle of the bond between the alpha carbon atom and the carbonyl carbon atom is represented by symbol psi. These angles are measured in degrees, where 180 degree is the angle of the bonds where all of the atoms of both residues lie in the most extended conformation. Rotation in one direction is positive, so the values go from 0 degree to 180 degree, and in the other direction they go from 0 degree to minus 180 degree. Hence, 180 degree is equal to minus 180 degree in this notation. These two angles define the conformation of that residue in a protein and are called the Ramachandran angles, which are now psi and phi. Most of the amino acid residues in a given protein are found in some form of secondary structure, such as an alpha helix, beta strands, or turns. The phi and psi bond angles for each residue in the alpha helical structure are very similar as shown on the left. This is why the structure is so regular. Similarly, the phi and psi bond angles for every residue in a beta strand are similar. Since the residues in an beta strand are in an extended form, the phi and psi angles in this conformation are close to 180 degree. For any given protein, you can plot all of the bond angles for every pair of residues. These can be plotted on a diagram called a Ramachandran plot, named after the biophysicist G. N. Ramachandran. Such a plot shows that most of the residues beta strands have similar bond angles that cluster in a region near the top left-hand corner of the diagram. Similarly, residues in a right-handed alpha helix have a very similar bond angles around psi 45 degree and 5 plus 45 degree. The residues in type 2 turns also have very characteristic bond angles. Some regions of the Ramachandran plot will be empty because of steric clashes between the oxygen atoms. These regions are mostly located in the lower right-hand corner of the plot. Now let me discuss what are torsional angles. Torsional angles are dihedral angles which are defined by 4 points in space. In proteins, the earlier mentioned angles, which were phi and psi. These are the two torsional angles which describe the rotation of the polypeptide chain around the two bonds on both sides of the carbon atom. The standard IUPAC definition of a dihedral angle is illustrated in the figure. A, B, C and D illustrate the position of the 4 atoms used to define the dihedral angle. The rotation takes place around the central BC bond. The deviation of the A' bond B and C bond D bonds from each other is measured by the deviation of D from A, that is rotation around the BC bond. Positive angles correspond to clockwise rotation. The Ramachandran angles in proteins are restricted to certain values, since some angles will result in sterical clashes between main chain and the side chain atoms in the polypeptide. In addition, for each type of the secondary structure elements, there is a characteristic range of torsion angle values, which can clearly be seen on the Ramachandran plot. Coming to next topic, which is DNA supercoiling. Cellular DNA is extremely compacted, implying a high degree of structural organization. The folding mechanism not only must pack the DNA but also must permit access to the information in the DNA. Supercoiling means the coiling of a coil. A telephonic cord, for example, is typically a coiled wire. DNA is coiled in the form of a double helix, with both strands of the DNA coiling around an axis. DNA supercoiling is generally a manifestation of structural strain. Perhaps less predictable is that replication and transcription of DNA also affect or are affected by supercoiling. Both processes require a separation of DNA strands, which is a process complicated by the helical interwinding of the strands. This indicates that supercoiling is an intrinsic property of DNA tertiary structure. It occurs in all cellular DNAs and is highly regulated by each cell. For circular DNA molecules, a topological property is one that is unaffected by deformations of the DNA strands as long as no breaks are introduced. Topological properties are changed only by breakage and rejoining of the backbone of one or both DNA strands. Linking number is a topological property of double-stranded DNA because it does not vary when the DNA is bent or deformed as long as both DNA strands remain intact. For a closed circular DNA molecule to have a topological property such as linking number, neither strand may contain a break. The linking number in relaxed DNA, which is represented by LK0, is used as a reference for the molecule. LK0 is equal to 200 if 2 turns are removed from this molecule when linking number equals 198. It is often convenient to express the change in linking number in terms of a quantity that is independent of the length of the DNA molecule. This quantity, called the specific linking difference or superhelical density, is a measure of the number of turns removed relative to the number present in relaxed DNA. Linking number can be broken down in two structural components, which are twist TW and width, which is WR. These are more difficult to describe than linking number, but width may be thought of as a measure of the coiling of the helix axis, and twist as determining the local twisting or spatial relationship of neighboring base pairs. When the linking number changes, some of the resulting strain is usually compensated for by width in supercoiling and some by changes in twist, which gives rise to the equation Tw, which is twist, and Wr, which is width, need not be integers. Twist and width are geometric rather than topological properties because they may be changed by deformation of a closed circular DNA molecule. Coming to the final objective, which is ligands and their interactions. In each cell of an organism, a myriad of reactions, covalent and non-covalent, occur at any given point in time. These reactions are coordinated and regulated both spatially and temporally. The term ligand in biological systems can have many different meanings. It is usually, in its broadest sense, used to mean any molecule which interacts with a given molecule, which in this case can be a protein. The term ligand includes other macromolecules, for example, peptides, proteins, nucleic acids, lipids, carbohydrates or mixed molecular species thereof, as well as small molecular mass arbitrarily that are less than 7 to 2 kilodalton molecules. Ligands comprise a very large and structurally diverse group of molecules which unsurprisingly also display a wide variety of physical chemical characteristics. Proteins play a pivotal, indeed essential role in cellular and in multicellular organisms, extracellular activities, etc. Proteins-ligand interactions play a key role in cellular metabolism. Detailed knowledge of such interactions at both a microscopic and a macroscopic level is required. Enzyme-ligand reactions form a very large group of important complexes which have been investigated for many years. In these systems, not only is the substrates of obvious importance, but so are other molecules which regulate enzyme activity such as coenzymes and positive and negative modulators in both the allosteric and non-allosteric proteins. Other important systems which are now being studied at a molecular level include ligand binding to structural proteins, protein DNA binding, polysaccharide, protein-protein interactions, and peptide-protein interactions. The formation and maintenance of the quaternary structure of multi-subunit proteins, the self-assembly of large structures such as microtubules and chromatin transcription factors, various promoter interactions are other examples, although the list is actually almost endless. With this, we conclude our today's topic. Hope you enjoyed and understood it well. Thank you.